All right. So we've finished discussing different antibacterial drugs, the resistance of microorganisms to those drugs, and I don't think there is any better example of antimicrobial resistance, resistance to antibiotics, is Mycobacterium tuberculosis. So we're going to chat about this microorganism for a little bit. So I'm going to mention two species here, Mycobacterium tuberculosis and Mycobacterium avium. You can see here the colonies of Mycobacterium tuberculosis, and they look somewhat like fungal colonies. That's probably the word where the word mycobacterium is coming from. Myco means, fungus. So Mycobacteria in general, and Mycobacterium tuberculosis specifically, pretty slowly growing microorganisms. Generation time for some Mycobacteria can be up to 2 weeks. For Mycobacterium tuberculosis, it usually ranges in a matter from days to a week, which makes growing this pathogen in the lab an extremely slow and painstaking task. As you may remember, the actual structure of the cell wall of Mycobacterium tuberculosis is gram-positive. So it has a membrane, okay, and outside of the membrane it has a cell wall. So that's typical gram-positive. But it turns out that outside of the cell wall, Mycobacterium tuberculosis has a layer of mycolic acid, which makes it somewhat separate from the rest of gram-positive microorganisms. Because first of all, you cannot stain Mycobacterium tuberculosis using a traditional gram stain. It just won't penetrate mycolic acid. So in order to stain them, you need to use what is called acid-fast approach. Sounds really fancy, but actually it's like gram staining, just different reagents. You also have like 3 different steps, and bacteria appear red under the microscope. Non-acid-fast bacteria appear blue. Mycolic acid provides several important advantages to Mycobacterium tuberculosis. First, it makes it quite resistant to drying. Second, it allows microorganism to survive inside of the phagocyte, inside of different phagocytes, especially macrophages. As you may remember from other classes, phagocytes in general and macrophages specifically are the cells that consume bacteria, debris, all the stuff that should be present in the human body, and they destroy it. Turns out when macrophage phagocytoses Mycobacterium tuberculosis, instead of killing it, instead of dying, Mycobacterium tuberculosis establishes an infection, a long-term infection of the macrophage. Okay? For instance, one of the mechanisms of killing bacteria is acidification of the phagosome, meaning that phagosome essentially is filled with acid. Mycobacterium tuberculosis produces enzymes that prevent this, so phagosome never becomes acidic, and that allows bacteria to survive. Does that make sense so far? Now, here's what happens during the infection. Usually, as you probably know, the main route of transmission, like overwhelming route of transmission for Mycobacterium tuberculosis, is respiratory, from person to person. Okay? So when bacteria enters the alveoli, okay, it forms granuloma. Why does it form granuloma? Here's what happens. Bacteria infects alveolar macrophages, and alveolar macrophages, upon the infection, start to produce a variety of inflammatory molecules that attract other human immune cells, like T cells, neutrophils. So far, does that make sense? But it turns out that cytotoxic response that is stimulated by the infection, it does lead to the necrosis of the tissue, but it does not destroy the bacteria, because bacteria are hidden where? Inside of the macrophages. Makes sense, right? So that ongoing necrosis and ongoing inflammation eventually lead to the formation of the barrier composed of immune cells and fibrin coating, and basically it's called sometimes a granuloma with a necrotic core. So that's necrotic core. That's the coating. This granuloma, step number 2, over here. Can you see that step number 2? This granuloma is basically not infectious. Microorganism is completely hidden, is completely, how should I say it? Its spread is limited. Its spread is actually non-existent because it's kind of imprisoned inside of that granuloma. And people may carry the tubercle, the granuloma, for an extended period of time, but at some point, granuloma breaks. Maybe due to the, usually due to the lowered status of immune system. Immune response cannot hold the bacteria back. When such tubercle breaks, microorganisms get back into the bronchioli, get back into the respiratory system, and the person can transmit the infection. Does that make sense so far? We're good? Now, what are the important consequences of this specific mechanism of persistence and spread? Think about this. Person gets infected. At some point, person can be Mycobacterium tuberculosis positive without necessarily spreading the infection to other people. Does that make sense? Also, this person can be TB positive for an extended period of time, and then later, maybe years later, become a patient with fulminant tuberculosis, spreading the infection to other folks. Okay, we're good so far? Now, in terms of epidemiology, it turns out that, first of all, it is very contagious. People are infected very easily, but only 5% people that are infected develop an actual clinical tuberculosis. Conservative estimates put about a third of the world population in the group of folks who are infected or have been infected. Does that make sense? Maybe they now cleared. So what facilitates the transmission of tuberculosis? Overcrowded places with poor sanitation, especially poor air quality, poor air circulation. Places like prison are especially at high risk. If you would look up something like, tuberculosis in Russian prisons, it's a huge problem. Virtually every person that has been imprisoned in Russia leaves the prison with tuberculosis, okay, because of the overcrowdedness, essentially. As far as I know, in the United States, for people who serve time in prison or for people who work in prison, the guidelines on the TB test are different from the general population because they are considered to be at the higher risk of being exposed to Mycobacterium tuberculosis. Does that make sense? Now, in terms of the overall death toll, last time I checked, tuberculosis is the #1 killer in the world. Every year, 1.7, among infectious diseases. Every year, 1.7 million people die of tuberculosis. Out of those, 400,000 also have HIV. And we're going to chat about it a little bit. So what are the types of Mycobacterium tuberculosis infection? So, primary tuberculosis upon the infection is usually asymptomatic, or it may manifest as the infection with fairly mild fever, virtually no cough, okay? And then it may go away because bacteria will end up in that tubercle. Now, if immune status is lowered or nutritional status is low, then primary tuberculosis may lead to the chronic infection. And in this case, bacteria start to drain from the tubercles in the upper respiratory tract. And the person becomes infectious and can transmit the disease to other people. The symptoms of chronic tuberculosis are described in all kinds of books, in literature, in fiction books. People cough. Cough is associated with sputum that is either green colored because it's pyogenic infection, or sputum can be bloody, night sweats, fever, weight loss. When you go to the physician and you answer that questionnaire, and they have questions, do you experience night sweats? Do you experience fatigue? Do you experience weight loss? Do you experience chronic cough? They specifically look for Mycobacteria tuberculosis. Now, this is the typical X-ray of the patients with the primary tuberculosis infection. Here you see a tubercle and lobar inflammation. Severe pulmonary tuberculosis is shown on the left. You see significant obstruction of the left lung. Now, first of all, before we move on to extrapulmonary tuberculosis, tuberculosis is treatable. Okay, the infection is very resistant, so drugs has to be selected. Usually it's a course, often it's a combination of the drugs, but it's absolutely treatable. Okay, if it is left untreated, tuberculosis may end up being extrapulmonary, side of the lungs, or another term for it is miliary tuberculosis. Miliary comes from the French word milieu, which means environment. So it gets into the internal environment of the body, extracellular fluid, and can affect virtually any organ. From the heart, I don't even have it in the list, but heart tuberculosis causes myocarditis, pericarditis. Very commonly gets into the lymph nodes. That's, sort of okay if it's called kidneys. Bones and brain. So brain and meninges is practically a death sentence. Same goes for the bone tuberculosis. So we had an experience, I probably mentioned it with my son having osteomyelitis. When lab results came back and his physician, his ID physician, looked at them, the only thing that he was looking for, it's not TB. Because if it's not, then we know what to treat him with. If it would be TB, that would be a totally different story. Okay? Because it's really hard to get antibiotics to the bone in the significant concentrations. Now, if tuberculosis is limited to the lymph nodes, you can cut them out. Sometimes it can get out of the lungs, get into the lymph node, form granuloma there, can be surgically removed. It's actually quite convenient. Now, you'll know the diagnostics. We do TB test quite routinely. So basically the idea is intradermal administration of tuberculosis antigen. If you have been exposed to tuberculosis in your life, not just exposed, if you had an infection, maybe asymptomatic infection, maybe you have cleared it, nobody knows. But if you have been exposed, your immune system can now recognize that same antigen. It's injected into the skin. Immune cells in the skin will present that same antigen to your immune system. Your immune system will produce that cytotoxic response. And that cytotoxic response will lead to the bump, the TB reaction on the skin. Does that make sense? There are methods, pretty routine clinical approaches to do the PCR, okay, to detect bacterial DNA in the blood. It's kind of a secondary. Chest X-ray and stain of the sputum. Actually don't have a picture of the stain. Now, why do we do so many steps? Because of this. BCG vaccine. The only thing that I remember from how to decipher the acronym BCG, B stands for bacillus. C and G are last names of two French dudes. So what it is? It's the Mycobacterium tuberculosis that was killed. Okay? So the idea is that if you vaccinate a person with BCG vaccine, you prevent tuberculosis. This idea didn't really work. BCG vaccine is used in quite a lot of countries all over the world, but the purpose of this vaccine is not to prevent tuberculosis, but to reduce the transmission of tuberculosis in children and to reduce the number of extrapulmonary cases. So this vaccine is not effective. Its efficiency is very limited. And since its efficiency is very limited, it is not used routinely in the United States. Okay? So unless, I don't know, in the military do they do tuberculosis? Okay. Yeah. So, and this is done basically if you get into another country and you get exposed there. In Russia, every person, every child is vaccinated against tuberculosis. Okay, we all get BCG vaccine. It's actually pretty mild, so you don't have any serious adverse reactions. But it becomes kind of a nuisance when you come here into the United States. Because if your immune response to that BCG vaccine when you were a kid was good, then when you go through the TB test in the US, you come out positive. And then you have to, I don't know, now what I've heard, they kind of ease it down. They said, you know, you basically feel out that, yeah, you were vaccinated and it's okay. But about 10 years ago, you had to go through the chest X-ray. And if they found it sort of not convincing, you would do prophylactic antibiotics. Just in case. Okay. The best prevention, actually, public health measures. Meaning, you know, avoid things being overcrowded. If people get sick, give them proper treatment so the infection is cleared, not, you know, shit antibiotics that will just make it the bacteria resistant. Now this is a really interesting map, I think. It shows you the incidence, cases per year in 2017. On the left, you see the legend, number of cases per 100,000. Okay, now, we all know that this distribution is, you have to take this data always with a grain of salt. Take this presentation of data. For instance, look at United States and Canada. 0 to 24 cases per 100,000 people. Now, here's the deal. If you have 1 case per 100,000, it will add up to, what? 3,000 TB cases a year in the United States. That's not a lot. If you go to 20, that ends up with 60,000 cases. They all fall in the same category, so I don't really know how many cases. And that's a huge difference. Does that make sense? Now you can see, we would look at the countries that are affected the most. I'm going to go to African countries last. So we've got Mongolia. I would say maybe because the country moves quickly from being completely agrarian country with cow herders into being more city, more urbanized. But so people don't really get vaccinated yet, but they already live in the large groups. Does that make sense? So they kind of start moving closer without proper sanitation measures yet implemented. Countries in the Southeast Asia, you see India, Pakistan, Afghanistan. India there are a lot of people there, like really a lot. It's just crowds. Pakistan and Pakistan, a lot of people there too. Afghanistan, probably lack of proper healthcare system. Southeast Asia in general, explosive growth of the urban population. Central and South Africa. The answer is poverty and HIV. Think about this. HIV really depresses the immune system, which makes fulminant tuberculosis more likely to develop, right? Which means the transmission is more likely to happen. If we would be able to eliminate HIV in those countries, tuberculosis epidemic would also go down. But HIV treatment is extremely expensive. So there's no way we're looking at elimination of HIV in Africa in, reasonable future, which means HIV and tuberculosis will coexist. If you would actually look at the numbers, I mentioned tuberculosis is 1.7 out of this 1.7, 400,000 cases of both HIV and TB. Second biggest infectious disease killer in the world is HIV. 1.3 million. Okay, HIV sort of alone. Okay. Does that make sense? Any questions? Now, you may have noticed one another species, Mycobacterium avium. It's the disease that is pretty common in birds, and it's the infection of bird digestive tract. So people pick it up from dealing with, bird feces for some reason. This infection affects exclusively people who are immunocompromised. People who are healthy, people who are immunocompetent, they are not at risk for Mycobacterium avium infection at all. Does that make sense to you? Questions? Good. Now, the last thing that I wanted to chat about is how we test the efficiency of antimicrobials. How we know which antimicrobial is to use. One approach is already familiar to you. It's called Kirby-Bauer disk diffusion test, shown here. So in this exemplary picture you see Pseudomonas aeruginosa on the left, Staphylococcus aureus on the right, and 4 discs, the same type of antibiotic, are applied to each plate. You can see that Pseudomonas is significantly more resistant than Staphylococcus aureus. Does that make sense to you? Because you basically look at what you're looking at. You're looking at the disc with a particular concentration of antibiotic in that disc. Antibiotic starts to diffuse away from the disc into the agar, and as it diffuses, its concentration gradually decreases. And bacteria, you can see bacterial growth at the concentration of antibiotic. That is, its minimal inhibitory concentration. The only problem with Kirby-Bauer, well, not only problem, but the problem with Kirby-Bauer disc diffusion test is that you don't really know what is the minimal inhibitory concentration is. Does that make sense to you? All you can say is, if I take antimicrobial 1 in the concentration so and so, the inhibition zone is going to be, I don't know, 5 millimeters. Okay? That's it. If I take it in the larger concentration, you have to make a new disc. You see what I'm trying to say? So, discs have fixed concentration. You can't really determine what is called minimal inhibitory concentration. What if you want to do that? Well, in this case, you have to take a different approach, which is dilution test. So look at the image number 3. Here you see 5 solutions with different concentrations of antimicrobial drug, from 2 to 32 micrograms per milliliter. You see those are 2 times diluted. So we go from 32 to 16, from 16 to 8, from 8 to 4, and 4 to 2. Okay, so those are serial dilutions of antimicrobial drugs. So far, are you with me? You're following? Okay, good. And then you inoculate these solutions with the bacteria of interest, and you look at the growth. So you can see that 8 microgram per mil and above inhibit bacterial growth. Does that make sense? Which tells you that your minimal inhibitory concentration, lies somewhere between 8 and 4 micrograms per milliliter. You didn't test 7 or 6 or 5, maybe they would still work. Does that make sense? You can test those. This dilution method allows you to determine the range in which your minimal inhibitory concentration lies. And you can narrow down this range if you want. So far, does that make sense? Now you can take this approach one step further and prepare serial dilutions of different antibiotics, like in the picture number 2, in the 96 well plate. So for instance, the first row, the row A, represents serial dilutions of clindamycin, and for that particular bacteria, clindamycin does not inhibit its growth in the range tested. Okay, minimal inhibitory concentration is more than 32. For penicillin, the inhibition occurs at the concentration of 0.06. Okay, so that's minimal inhibitory concentration for penicillin. For erythromycin, minimal inhibitory concentration will be 8 micrograms per mil. So you can test multiple antibiotics, does that make sense, on one plate against single bacteria, okay, and get your answer pretty quickly. Okay, we're good so far? Good. The combination of the dilution test and Kirby-Bauer disc diffusion test is called E-strips. So in this image, number 4, you can see a vancomycin strip and the concentration of antibiotic that is infused sort of into the strip, you can see it decreases from 256 to 0.060. So the concentration decreases, the inhibition zone size becomes smaller and smaller. You see that? So in this case, you can determine the minimal inhibitory concentration, and the method is also very visual. You can do it using the plate, the agar. Does that make sense to you? Okay. Now, do you understand what these methods are? So the questions will be either match the method and the description, and you also have to know which method allows you to determine minimal inhibitory concentration. Basically, all dilution methods allow you to do that. Kirby-Bauer, disc diffusion doesn't. Okay. Now, the last thing I wanted to discuss is the therapeutic index of a drug, and I want to apply this specifically to antibiotics. So what is therapeutic index? For this we need to discuss what is the median effective dose and median toxic dose are. ED50 is the median effective dose, meaning that at this dose, if you treat a cohort of patients, say of 100 people, in 50 patients you will see a recovery. So effective dose is the dose of a drug at which this drug is effective in 50% of patients. Does that make sense? Does it make sense to you? Now, median toxic dose is the dose that causes toxicity in 50% of patients. All right? So therapeutic index here is the ratio between the median toxic dose and median effective dose. So let's play with this a little bit. If you have a drug which median effective dose is 1 and median toxic dose is 10, what's the therapeutic index? Come on. 10. Yes. Now, you have second drug, median effective dose is 1 and median toxic dose is 100. Therapeutic index is 100. Which one's better? First, second. Second is better. Why is it better? Yes. You have more flexibility in using higher doses. Okay? So for instance, if your current dose doesn't work, you can increase the dose, and you can increase the dose, and you can increase the dose. Does that make sense? So you have more leverage against the condition. Now, I'm not aware of any drugs that will have therapeutic index less than 1. But there was a story when some antiparasitic drug was developed for veterinary use, and it was tried in horses, and they gave it to a horse in the evening, and they tested the concentration of number of parasites in the blood, like 2 hours later, and it was declining, so it was definitely pretty effective. And then next morning the horse was dead. So that was a drug of therapeutic index 1, when effective dose and toxic dose were essentially the same. Okay. We're not using them, of course, but kind of give an idea. So what you should be able to do, you should be able to calculate therapeutic index based on the doses that I give you. Am I clear? I'm not going to give you any complicated numbers. It's going to be easy. And you have to be able to tell me which drug is better based on the therapeutic index for the drug. We're good?
So the higher the better.
Of course. Always. Yep. The higher the better. Got it? Okay.